And so this is what's referred to as the central dogma of molecular biology, where you have the DNA, which has the instructions. Then there's an intermediate called the RNA, which is really just a copy of a portion of the DNA. And then that encodes for a protein, which has a three-dimensional shape. And so by analogy, if you think of an instruction book that has the instructions for making all of the different parts of the car, that's like the DNA. And then the RNA is a copy of one page, so maybe a photocopy of the instructions for making a wheel. And then the wheel is a protein that has a specific function.
Sometimes this mutation in the genome can lead to a misfolded protein. So genetic variants can have a whole range of how common they are. They could range from being present in one person in the entire population of 7 billion people, or they're present in half the people in the population. And genetic variants can have a wide range of effects. So if I say the green car here is normal, you could have the vast majority of genetic variants in the population, 99.9% of them are also completely benign or have really tiny effects. So you can imagine a genetic variant that changes the color of the car from green to blue, has absolutely no consequence for the function of the car. And some genetic mutations are actually beneficial for you. They can make this car go a little bit faster or something like that. And there's also some genetic mutations that are damaging. This often occurs in the context of disease. So in the rare monogenic, the name suggests that monogenic means single gene. So it's usually a single mutation of really large effect. If you have the mutation, you're going to have disease. If you don't have the mutation, you won't have the disease. And here the environment's less important. So no matter what diet you eat or how much you exercise, if you have the mutation for sickle cell anemia, you're going to get sickle cell anemia. Your environment's much less important. So examples will be, as I mentioned, cystic fibrosis or sickle cell anemia, Duchenne's muscular dystrophy. These are all really rare, debilitating diseases with really early onset morbidity for most people with the disease. And then there's common polygenic diseases. So these are usually more common diseases, and polygenic just means poly means lots. So lots of genes are involved. And here there's many genetic variants, many genes, many mutations in many genes, oftentimes thousands of genes that are involved in the disease. And each of these genetic mutations confers a tiny bit of risk to that disease. But added together, they can have a substantive increase in risk. And here the environment's often very important for whether or not you get the disease or not. So genome-wide association, or GWAS, it's a large study that you perform in many numbers of people. You do this in general population to understand what genes contribute to the variation in the general population of LDL cholesterol. And because you have so many environmental contributions, each gene only contributes a small amount of risk. You need many people to find these genes. We're going to find the genes for this kind of disease in a linkage study. We're going to recruit families where we find many individuals with this disease. And so we're going to find which mutations are linked to disease. Again, this is a linkage studies. We're going to find out which mutations are present in only the people who have the disease and not in the people who don't. And because the genetics, a single mutation of large effects can cause you to have disease.
There's a small number of people that have a mutation that completely inactivates the PCSK9 gene so that it's not active. And it turns out that these people have much lower, on average, blood cholesterol LDL levels. And again, this greatly reduces their chance of heart attacks. If the mutated PCSK9 cannot eat the LDL receptors, there's more LDL receptors on liver cells. And that means that more LDL can be taken out of the bloodstream into the liver. And so pharmaceutical companies recently, after this finding, became very interested in this and thought, what if we could use our knowledge of genetics of these people, these rare people who are naturally lucky in that they have a much reduced probability of heart disease, and we can actually design a drug that mimics that effect. And so what they wanted to do is see if they can, in people who have the normal PCSK9 that can degrade LDL receptors, see if we can block it. And so what two pharmaceutical companies have done, and these drugs have finished pretty much almost all of the clinical trials and will likely be available in the next year or two in the clinic, is they made drugs that block this protein. So what happens is the drug actually physically binds to PCSK9 protein and blocks it such that the protein cannot chew up LDL receptors. And so it turns out this drug is very effective. It, again, mimics this natural mutation, and it turns out in these clinical studies it was shown that it's better than any other cholesterol lowering drug.